This video covers a sample MCAT passage walkthrough. The passage focuses on ions, paramagnetic, and diamagnetic species. Let's start. Bacterial biofilms pose a significant clinical risk and are difficult to eradicate. To achieve precise control over biofilm behavior, researchers developed a bioelectronic ion delivery system to regulate the growth of Bacillus subtilis biofilms. Figure 1. In this system, a pulse voltage was applied across AgCl electrodes immersed in a KCl reservoir. The applied electric field drives K plus ions through an anionic hydrogel and into the agar substrate containing the biofilm. The localized delivery of extracellular K plus was monitored using IPG4, which is a fluorescent potassium indicator. Here's figure number 1. It says bioelectronic ion delivery system. Under resting conditions, B subtilis maintains a high intracellular K plus concentration of about 300 millimolar relative to the extracellular environment about 8 millimolar, elevating extracellular K plus near the working electrode WE region to about 150 millimolar disrupts this gradient and induces opening of potassium channels, leading to changes in membrane potential. These changes were measured using thioflavin T, THT, a positively charged membrane permeable dye whose intracellular fluorescence increases as the membrane becomes more negative. The resulting membrane potential changes are shown in figure 2. Here's figure 2, and it says THT fluorescence observed at the WE region exposed to bioelectronic pump and the dormant region not exposed to bioelectronic pump. Here's question number 1. In a photoionization experiment, potassium metal undergoes the following reaction. K plus HV, which is light or energy, goes into K plus electron, plus one electron. If the metal ion product of this reaction undergoes another round of photoionization, which of the following graphs best represents the energy profile for this second step? Here is question number 2. It reads, which of the following best explains the result presented in figure 2? Answer choice A says a decrease in the extracellular electric field increases THT uptake, leading to increased fluorescence at the WE region. Answer choice B says the THT induced fluorescence observed in the dormant region is due to the increased electric force generated. Answer choice C says the electric field generated indirectly hyperpolarizes the cell membrane, increasing THT fluorescence in the WE region. Answer choice D says an increase in electric force induces a hyperpolarized state, causing THT fluorescence to remain approximately constant in the dormant region. Question number 4. To identify the ions involved in the bioelectronic ion delivery system, mass spectrometry was performed. During analysis of one of the ions used to initiate the experiment, the ion was observed to be weakly attracted to an external magnetic field. Which of the following ions correspond to this observation? Here is question number 5. It says which of the following amino acids lines the inside of K plus channels in B subtilis cells at physiological pH? Let's go over this passage and analyze it. Having a strong passage map is important because it allows us to predict, at least to some degree, what kind of questions might be asked. It also helps us to focus our attention on topics that may be tested. So let's begin. The first sentence reads: Bacterial biofilms pose a significant clinical risk and are difficult to eradicate. The first sentence introduces bacterial biofilms and states that they are difficult to eliminate. What do we know about biofilms? They are structured communities of bacteria that can withstand many treatments that are effective against free floating bacteria. From this first sentence, we can predict that the passage will likely discuss bacteria and possibly novel methods to control or eliminate bacterial biofilms. The topic of bacteria is very broad, so at this point, we do not need to review all bacterial biology. Instead, we should make a mental note to expect questions related to biofilms or bacteria. Let's move on to the second sentence. Second sentence reads: To achieve precise control over biofilm behavior, researchers developed a bioelectronic ion delivery system to regulate the growth of Bacillus subtilis biofilms. Figure 1. The second sentence confirms our prediction. The author introduced a novel method that allows scientists to control the growth of bacterial biofilms. They worked with Bacillus subtilis. This is a specific information that we should underline in case a question refers to it. The passage also introduces a bioelectronic ion delivery system. We are unfamiliar with this term, and this is completely fine. We should continue reading to understand how it operates and why it is important. Figure 1 is also provided to illustrate this system. The third sentence says, in this system, a pulse voltage was applied across AgCl electrodes immersed in a KCl reservoir. The third sentence explains that this bioelectronic system functions similar to an electrochemical cell, specifically an electrolytic cell, in which energy voltage is used to move ions in a controlled manner. The electrodes used are Ag and AgCl, and they are immersed in KCl solution. Although we could take the time to draw out the full electrolytic cell, that would be very unnecessary, unless a question specifically tests electrochemistry. For now, it is sufficient to make a mental note that this system behaves like an electrolytic cell. The applied electric field derives K plus ions through an anionic hydrogel and into the agar substrate containing the biofilm. This fourth sentence provides more details. As expected, the purpose of the electrolytic like system is to use voltage to control the movement of ions, specifically in this case, potassium K plus ions, and to direct them toward the agar substrate containing the biofilm through an anionic hydrogel. The mention of an anionic hydrogel may be important. Even if you are unfamiliar with hydrogels, we should recognize that anionic means negatively charged. This makes sense because negatively charged material would allow positively charged K plus ions to pass through, whereas a positively charged material would repel the positively charged K plus ions. Figure 1 illustrates this setup described in this first paragraph. At this point, we should ask ourselves why the researchers are interested in K plus ions and how their movement might affect biofilm growth or their control. Formulating these types of guiding questions helps us to better understand the passage. Let's move on to the fifth sentence. The fifth sentence reads: The localized delivery of extracellular K plus was monitored using IPG4, which is a fluorescent potassium indicator. The fifth sentence states that the researchers fluorescently detect the localized delivery of K plus ions using a potassium sensitive fluorescent indicator. This still does not explain why potassium is important, but it tells us that K plus ions are delivered to the extracellular fluid surrounding the biofilm. We should continue reading to gain more information. The sixth sentence reads, under resting conditions, B subtilis maintains a high intracellular K plus concentration of about 300 millimolar relative to the extracellular environment of about 8 millimolar. This sixth sentence provides additional details. It explains that the concentration of K plus ions is normally higher inside bacterial cells. The seventh sentence reads, elevating extracellular K plus near the working electrode WE region to about 150 millimolar disrupts this gradient and induces opening of potassium channels, leading to changes in membrane potential. This following sentence states that increasing extracellular potassium concentration leads to the opening of potassium channels, which alters the electrochemical gradient across the cell membrane and it changes the membrane potential. Now we understand why potassium is important to some extent. Delivering K plus ions using the bioelectronic system changes the membrane potential, which may affect how the biofilm functions. The next sentence reads, these changes were measured using thioflavin, THT, a positively charged membrane permeable dye whose intracellular fluorescence increases as the membrane becomes more negative. The resulting membrane potential changes are shown in figure 2. So the passage then explains that a fluorescent dye called the THT is used to monitor changes in the electrochemical gradient. To understand this, we need to follow a sequence of events. First, a voltage pulse controls the movement of K plus ions. The voltage pulse creates an electric field which deposits K plus ions on the extracellular side of bacterial cells within the biofilm. The increased extracellular K plus concentration influences potassium channel activity. When the potassium channels open, K plus ions move out of the cell down their concentration gradient because their concentration is normally higher inside the cell. As positive ions leave the cell, the interior of the cell becomes more negatively charged, altering the membrane potential and causing the cell membrane to become hyperpolarized. This increased negative membrane potential allows the positively charged THT fluorescent dye to enter the cell, enabling fluorescence detection. The passage says that the result of this experiment are presented in figure 2. The main idea of the passage is that the passage describes how a bioelectronic ion delivery system manipulates K plus ion gradient to alter membrane potential and regulate bacterial biofilm behavior. This sample MCAT passage is a great example of how the topic of ions can be presented within an MCAT passage. As we saw, ions appear in multiple contexts in this passage. For example, they appear in electrolytic cells. They appear as individual ions, such as K+, in electrochemical gradient across cell membranes, and in ionic compounds such as KCl and AgCl. The topic of ions is a very broad topic and can be tested in many different contexts. Let's now move on to the questions and see how we can answer the questions. Here's question number 1. It read, in a photoionization experiment, potassium metal undergoes the following reaction. If the metal ion product of this reaction undergoes another round of photoionization, which of the following graphs best represents the energy profile for this second step? The answer to this question is D. Let's break down why. First, let's ask ourselves, what is this question really asking us in simple terms? The scenario describes potassium metal undergoing photoionization, in which it loses one electron and becomes a potassium cation, K+. This question is simply asking us to match a graph to an endothermic process. The graphs provided are energy versus reaction progress diagrams. We are asked to match the correct graph to the second ionization process, which we would recognize as an endothermic process. And how do we know this? From our background knowledge, elements in group 1, such as potassium, readily lose one electron to achieve the electron configuration of their nearest noble gas. After losing one electron, potassium attains the electron configuration of argon, which is especially stable. Removing an additional electron from this already stable potassium cation requires a significantly larger amount of energy. Therefore, the second ionization energy is much higher and it involves overcoming a large activation energy barrier. Because energy must be absorbed to remove this second electron, the process is endothermic. In an endothermic reaction, the products are at a higher energy than the reactants. This means energy is taken in during the reaction. The energy profile that starts lower and ends at a higher energy is graph D, making graph D or answer choice D the correct answer. Graphs A and B represent exothermic processes in which the reactants start at a higher energy than the products, indicating that energy is released. Graph C is not correct because the reactants and the products are at the same energy level, suggesting no net energy change. This is a classic example of how the MCAT tests your understanding of ions and electron configurations. Always read the question stem carefully, break the scenario down into simple terms, and then apply the fundamental concepts that you have previously learned to arrive at the correct answer. Let's move on to question number 2. Question number 2 reads, which of the following best explains the result presented in figure 2? This question is asking us to analyze figure 2 and determine which answer choice best explains the result presented. We should refer directly to figure 2 to answer this question. From figure 2, we can see that THT fluorescence increases over time in the WE region. The fluorescence in the WE region is represented by the bluish green bars. The WE region is located near the bioelectronic device, where K plus ions were deposited. In contrast, the dormant region, shown in pink, does not display a significant overall change over the 3-hour time frame. Although there is a slight decrease at the 2-hour mark, followed by a little bit of increase, there is no consistent upward trend. The dormant region is located farther away from where the K plus ions were deposited. The figure itself does not explain why this trend occurs. To understand the reason behind the result, we must refer back to the passage or our passage map. This is why having a strong passage map is very valuable. As we noted earlier, the bioelectronic ion delivery system functions like an electrolytic cell. The pulse voltage allows for the controlled release of K plus ions at the WE region. The increased extracellular concentration of K plus ions influences potassium channel activity and promotes K plus efflux, or the exit of potassium ions from the bacterial cells. As K plus ions leave the cell, the interior becomes more negative. In other words, the membrane becomes hyperpolarized because positively charged potassium ions are exiting the cell. This change in membrane potential allows the positively charged THT fluorescent dye to enter the cell, leading to an increased fluorescence observed in the WE region. Therefore, we must select the answer choice that matches this mechanism. Let's evaluate the options one by one. Option A says a decrease in extracellular electric field increases THT uptake, leading to increased fluorescence at the WE region. This is not correct. The passage does not describe a decrease in extracellular electric field. The applied voltage generates an electric field at the bioelectronic device, which directs K plus ions toward the extracellular side of the bacterial cells. This option does not accurately describe the mechanism. Let's move on to option B. Option B says the THT induced fluorescence observed in the dormant region is due to the increased electric force generated. This is also not correct. The bioelectronic system operates at the WE region, not the dormant region. The increase in fluorescence occurs in the WE region, not in the dormant region. Answer choice C. It says the electric field generated indirectly hyperpolarizes the cell membrane, increasing THT fluorescence in the WE region. This is the correct answer. The pulse voltage creates a potential difference, which generates an electric field. This electric field controls the movement of K plus ions, directing them to the anionic hydrogel and depositing them on the outside or the extracellular side of the bacterial cells. This process leads to potassium efflux and membrane hyperpolarization. The negatively charged membrane then allows the positively charged THC dye to enter the cell. It increases fluorescence in the WE region as shown in figure 2. So answer choice C is the correct answer. Answer choice D says an increase in electric force induces a hyperpolarized state, causing THT fluorescence to remain approximately constant in the dormant region. This is also not correct because the hyperpolarization occurs in the WE region, not the dormant region. This question is a great example of how the MCAT tests your broader understanding of an entire passage. Having a strong passage map allows you to connect the mechanism described earlier with the data presented in the figure and arrive at the correct answer more efficiently. When you encounter a question like this, rely on your passage map and apply the same structured reasoning process as we did for this passage. Here's question number 4. It reads to identify the ions involved in the bioelectronic ion delivery system, mass spectrometry was performed. During analysis of one of the ions used to initiate the experiment, the ion was observed to be weakly attracted to an external magnetic field. Which of the following ions correspond to this observation? So here is question number 4. What is this question really asking us? Let's simplify it into the most basic terms. The question is asking us to determine which of the listed ions is paramagnetic. From our background knowledge, we know that paramagnetic species are weakly attracted to an external magnetic field because they have at least one unpaired electron. Diamagnetic species, on the other hand, have all electrons paired and are weakly repelled by the magnetic field. How do we determine whether a species is paramagnetic or diamagnetic? We must write the electron configuration for each ion and identify whether any unpaired electrons are present. K plus has the electron configuration of argon. This is a full noble gas configuration with all electrons paired, so it is diamagnetic. Ag2 plus, however, loses 2 electrons. Removing 2 electrons from silver leaves a 4d9 configuration. A d9 configuration contains one unpaired electron, making it paramagnetic and therefore weakly attracted to a magnetic field. Ag plus loses 1 electron from its neutral silver atom, resulting in a filled 4d10 configuration. Since all electrons are paired, it is diamagnetic. Cl minus gains 1 electron to achieve the electron configuration of argon. Again, this configuration is fully filled and diamagnetic. Thus, the ion that would be weakly attracted to an external magnetic field is Ag2 plus. The correct answer is answer choice B. This question is a good example of how the MCAT may disguise topics such as paramagnetism, ions, and electron configuration within an experimental context. Let's move on to the last question. Question number 5. It reads, which of the following amino acids lines the inside of K plus channels in B subtilis cells at physiological pH? To answer this question correctly, we need to determine which amino acid is located inside a potassium K plus channel and allows the entry, or in this case, or in the case of this passage, the exit or the efflux of K plus ions from B subtilis cells at physiological pH. The passage does not mention any amino acid, nor does it provide any information about the structure of potassium channels. The only information given is that K plus ions leave the bacterial cells via this channel. So positively charged residues would repel K plus, while negatively charged residues would attract and stabilize K plus as it passes. Among the options, answer choice A, lysine, is positively charged and it would repel K plus. Answer choice B, alanine, is neutral and nonpolar and it does not interact electrostatically. Answer choice C, glutamic acid, is negatively charged at physiological pH and it attracts K+, which facilitates passage of K+ ions. Cysteine, on the other hand, is neutral at physiological pH and it does not strongly interact. Therefore, the correct answer is glutamic acid, answer choice C, because it is negatively charged at physiological pH and can be found inside the channel opening to help K plus efflux and leave the cell. This concludes the video. Thank you very much for watching and all the best luck to you.